And now, let's meet our fifth finalist, Leslie Heid from the Center for Complex Biological Systems.
I'm Leslie Heid, and I'm in the Mathematical, Computational, and Systems Biology PhD program. So I have the coolest part-time job. I work for NASA. I do mission modeling for their deep space network, which is the moon and beyond. I look at communication needs of different spacecraft for the next 40 years. So I'm doing things that are launching on the Artemis program, little CubeSats that are going up, all the way to the Mercury lander we're sending out in 2035. I model all of that, and it's a fantastic job. I love it. It's so interesting. What I do at NASA is completely different than the research I'm doing in Dr. Downing's lab. At NASA, I do a lot of research into the components of the spacecraft, of its communication systems, and I have to input all of these things into a very complicated system that they have that analyzes everything, and it's very complicated. That is more research and modeling based. What I do for Dr. Downing's lab with the methylation project is a lot of coding, because I take these simulated data sets and I analyze them, and we make the data sets resemble certain biological systems. And then I have to write these codes to see if I can organize all of the different DNA reads into an order that makes sense and that is usable, and that hopefully will turn into different organs and all sorts of amazing science in the future. I would not be able to go to school and have the part-time job and have an amazing daughter without my husband. He is my rock, and I am so blessed to have him take care of her while I'm studying my butt off. I have the perfect life.
Studying your butt off.
Yes, I studied my butt off.
And now, let's hear Leslie's pitch. Window of opportunity, quantifying DNA methylation pseudotime to rewrite a cell's fate.
Every once in a while, we unlock a potential so powerful it reaches out and changes humanity forever. In 1953, James Watson and Francis Crick identified the double helix structure of DNA from X-ray images taken by Rosalind Franklin. And in the 68 years since, manipulation of the genetic code has exploded into countless applications and industries. Genetically modified crops, mail-order DNA genealogy kits, new vaccines developed in less than a year to combat worldwide pandemics. And still, we have only barely begun to understand the staggering complexity of this tiny but immensely powerful molecule. Within each of us are trillions of identical copies of DNA encapsulated in the nuclei of our cells. Brain cells, liver cells, skin cells all have the exact same blueprints necessary to make every protein our bodies could ever need. But if every cell has the same master plan, why do they all act differently? Epigenetics and DNA methylation. As an embryo develops, it reaches a point where the cells begin to differentiate, to become a heart, a nervous system, a skeleton. Methylation is when methyl groups attach at various points along the DNA strand to either prevent or promote the expression of specific genes. For example, muscle cells don't need the dendrite structures of nerve cells, so those genes are suppressed. Once a cell has been methylated, the children of that cell are usually methylated the same way following each division. But what if they weren't? During mitosis, there's a narrow window of time where each resulting child cell is only half methylated from the half of the DNA that came from the parent cell. The newly added complementary DNA is unmethylated, but only until new methylase enzymes can attach. Now, what if we could stop the process, or even redirect it? It may be possible to essentially rewrite a cell's fate to become a different cell. The problem is, no one has been able to identify nascent cells at this key cell fate juncture. Current genomic sequencing technology cannot pinpoint this narrow window because their DNA libraries come from cells at varying stages in the cell cycle. That's where I come in. Determining the age of cells based on their position in a biological process is known as determining their pseudotime. No one has yet developed pseudotime based on methylation signatures. My research is to quantify methylation pseudotime in order to identify nascent DNA strands. Once identified, a whole host of new, targeted experiments can be performed with the ultimate goal of redirecting cells to perform different tasks. Someday, from a simple swab inside a patient's cheek, we could grow a replacement liver or kidney, or replenish bone marrow with a perfect match. My work will be a vital step in unlocking this potential, and once we succeed, humanity will be changed forever. Thank you.
Thank you, Leslie. And now, let's get to know you a little bit. Now, let's be honest, it's going to be hard to top NASA, but is there anything else that some people may not know about you?
There are actually two things that people don't know about me. The first one is that I have no Google foo, which is when I don't know something and I need to look it up online, I don't even know the right keywords to use to get the information that I want. I have no foo, no Google foo. I can't find anything when I look online. It's terrible. And the second thing is that I really want to dye my hair a crazy color. It's the pandemic. I'm at home. Nobody can see me except for right now. But I want to go blue or purple or something crazy. But I just haven't had the guts to do it yet. So
have you tried the filters to give you a taste of what it's like, and so you can sort of, in the virtual world, feel it out?
What do I have to Google in order to pick up one of those?
I see the problem. I'm understanding now. Never mind. We'll talk afterwards.
Oh, thank you.
And then if there's maybe one piece of advice you have for people thinking about grad school, what would that be?
Wow, that's a really good question. I would have to say, just do it. Because I have imposter syndrome, which is where I don't think I'm as smart as all these other people in my labs and classes. Like, they're brilliant and they know how to work all this complex equipment, and I don't know any of this stuff. But that's the thing. I'm here to learn. I don't know any of this stuff yet. So when I was applying for grad schools, I was like, am I smart enough? Am I good enough? And I'm just really glad that UCI gave me a chance. And I know that I'm going to study my butt off. And I will know all these things, and I will be good enough. Yeah.
Yes, absolutely. And I think we are all learning from you all tonight about the fabulous research that you're doing. So thank you for being here with us tonight.
Thank you.